Good evening. Today's lecture topic is biodiversity and conservation, that is plant conservation and biotechnology, especially related to in situ and ex situ conservation. This whole chapter is divided into the importance of the biodiversity, loss of biodiversity, then conservation of biodiversity, regulations, role of biodiversity in plant conservation, first in situ, second ex situ conservation. Biodiversity is degree of variation on life forms, that is flora and fauna. It is the totality of genes, species, and ecosystems of a particular region. Importance of biodiversity. Biological diversity is the very basis of human survival and economic development, as it provides food, housing, clothing, medicine, and industrial raw material. Second, a wide range of industrial materials are directly obtained from biological resources, such as rubber, oil, fiber, building material, timber, and paper. All are obtained from biological resources. 80% of the world population depends on medicines obtained from nature. Loss of biodiversity. The main causes of biodiversity loss are habitat destruction, invasive species, जिनको कि exotic species भी कहा जाता है, जैसे पार्थेनियम, आइकॉर्निया, लेंटाना, ये कुछ 2-3 ऐसी स्पीसीज हैं जो बाहर से आई और यहाँ उन्होंने हैवोक पैदा कर दिया. Pollution, population, over exploitation and climate changes. Then introduction of exotic species, as I have just told you. Second, destruction of the habitat is the biggest threat to biodiversity. Habitats can also be damaged by flooding, lack of water, climate changes, salination, etc. All phenomena which may be both natural and man-made. Third, due to global warming, also affects plants, animals, and microbes, both by changing their habitats and also by direct effects on temperature. Fourth, climatic changes also affect species at cellular level and can alter or change the genetic makeup of the cell. Now, conservation of biodiversity. First, by protecting the loss of biodiversity, both in situ and ex situ methods of biodiversity conservation are equally important. Second, it is now recognized that ex situ techniques can be efficiently used to complement in situ methods, and they may represent the only option for conserving certain highly endangered and rare species. This is a report of 2000 by Ramsey et al. Third, preserving the habitat is the most important issue in the conservation of biodiversity. Fifth, since chemical pesticides are also responsible for a large number of animal deaths occurring every year, minimizing the use of chemical pesticides is another way or technique for the survival of biodiversity. Next, conservation of biodiversity through establishment of protective areas like national parks, wildlife sanctuaries, biosphere reserves, and marine reserves, etc. Role of biotechnology in plant conservation. Biotechnology can be used in both the ways. First, in situ conservation, which include biosphere reserves, national parks, tiger reserves, and wildlife sanctuaries. Second, ex situ conservation, that is selection of superior germ plasm, field gene banks, botanical garden, and experimental gardens. Protective areas. The protective areas are constituted and governed or controlled under the provision of the Wildlife Protection Act 1972, which can be amended or changed from time to time as required with the changing ground realities concerning wildlife crime control and protective areas management. Then implementation of this act is further complemented by the other acts such as Indian Forest Act 1972, Forest Conservation Act 1980s, Environment Protection Act 1986, Biological Diversity Act 2002, the Scheduled Tribes and other traditional forest dwellers recognition of Forest Right Act 2006. So in order to strengthening the synergized global wildlife conservation efforts. India is a member of major international conventions or conferences. So by these, we can conserve our biodiversity. State-wise detail of the protective areas network of India. ये करीब 30 स्टेट्स के बारे में दिया हुआ है और हर स्टेट में नंबर ऑफ नेशनल पार्क्स, नंबर ऑफ वाइल्ड लाइफ सेंचुरीज, नंबर ऑफ कंजर्वेशन रिजर्व एंड नंबर ऑफ कम्युनिटी रिजर्व। जैसे आंध्र प्रदेश में आप देख रहे हैं कि 6 नेशनल पार्क हैं, 21 सेंचुरीज हैं। अरुणाचल प्रदेश में 2 हैं, सेंचुरीज 11 हैं। और इसी तरह आप देख रहे हैं कि उत्तर प्रदेश में 1 नेशनल पार्क और 23 सेंचुरीज है। ये कुछ एग्जाम्पल है स्टेट वाइज। फिर नेशनल पार्क की जो देख रहे हैं संख्या वो अलग अलग है। जैसे तमिलनाडु में दिखाया गया है, अलग अलग स्टेज वाइज दिखाने की कोशिश की है। अब हम आते हैं मेन टॉपिक पर, इन सिटू प्रोटेक्टिव एरियाज। तो इसको 4 ग्रुप में डिवाइड किया गया है, बायोस्फीयर रिजर्व्स, नेशनल पार्क्स, टाइगर रिजर्व्स एंड वाइल्ड लाइफ सेंचुरीज। Biosphere reserves क्या होते हैं? A biosphere reserve is an area proposed by its habitat ratified by a national committee and designated by UNESCO Man and Biosphere Program in 1971, which demonstrate innovative approaches to living and working in harmony with the nature. The term biosphere refers to all of the land, water, and atmosphere that supply life on Earth. The word reserve means that it is a special area recognized for balancing conservation with sustainable use. Each biosphere reserve demonstrates practical approaches to balancing conservation and human use of that particular area. Some of the important biosphere reserves in India are: फर्स्ट इज नीलगिरी बायोस्फीयर रिजर्व, जो कि तमिलनाडु, केरला और कर्नाटक को कवर करता है। 2000 में इसका निर्माण किया गया था। गल्फ ऑफ मुन्नार बायोस्फीयर रिजर्व, ये भी तमिलनाडु में है। सुंदरवन बायोस्फीयर रिजर्व, ये वेस्ट बंगाल में है। नंदा देवी, ये उत्तराखंड में है। नौटेक बायोस्फीयर रिजर्व, ये आपका मेघालय में है। पंचमढ़ी बायोस्फीयर रिजर्व आपका मध्य प्रदेश में है। सिमीपाल उड़ीसा में है। और इसी तरह अरुणाचल प्रदेश और अंडमान निकोबार में भी है। ये यूनेस्को की साइड है, वो इसमें समझाने की कोशिश की है। यदि बायोस्फीयर रिजर्व है तो उसको एरिया में डिवाइड किया गया है। दिस इज द कोर एरिया, जहाँ पे एनिमल्स और प्लांट्स इंपॉर्टेंट मिलते हैं। दिस इज बफर जोन। बफर जोन का मतलब है वो बीच का इलाका जो जंगल और आबादी के बीच का इलाका है। एंड दिस इज ट्रांजिशन जोन, एग्जैक्टली आपको एडजेसेंट टू द पॉपुलेशन। तो ऐसे बहुत सारे एरियाज हैं। अब आता है नेशनल पार्क के बारे में। नेशनल पार्क इज ए पार्क in use for conservation purposes. National park is in an area having adequate ecological, faunal, floral, geomorphological, natural or geological significance. The national park is declared for the purpose of protecting, propagating or developing wildlife, including flora and fauna or its environment. Although individual national designate their own national park, जैसे टाइगर रिजर्व्स है, वो भी बेसिकली नेशनल पार्क है। सो नेशनल पार्क्स आर ऑलमोस्ट ऑलवेज ओपन टू विजिटर। आप चाहे तो वहां पे घूम सकते हैं। बायोस्फीयर रिजर्व में ऐसा नहीं है, आपको परमिशन लेनी पड़ेगी। जैसे उत्तराखंड में एक जगह है वैली ऑफ फ्लावर, तो वैली ऑफ फ्लावर के लिए बिना परमिशन के लिए नहीं जा सकते आप। उसके लिए पास बनता है, फीस देनी पड़ती है। ऑब्जेक्टिव ऑफ नेशनल पार्क टू प्रोटेक्ट natural biodiversity along with its underlying ecological structure and supporting environmental processes, and to promote education and recreation. नेशनल पार्क वर्सेस वाइल्ड लाइफ सेंचुरीज में बेसिक क्या डिफरेंस है? इन ए नेशनल पार्क नो राइट्स आर अलाउड. Second, no grazing of any living. While in addition, from a national park requires recommendation of the National Board for Wildlife. सेंचुरी में थोड़ा सा फर्क है, वहां पे हमारे कुछ राइट्स हैं, जैसे मैंने अब क्लास में भी बताया था आपको कि बलिया में एक सेंचुरी है, जयप्रकाश नारायण बर्ड सेंचुरी, सुराताल, बसंतपुर, बलिया. Certain rights can be allowed in a sanctuary. The chief wildlife warden may regulate, control, or prohibit. While any removal of exploitation of wildlife or forest produce from a sanctuary requires the recommendation of the State Board of Wildlife. Wildlife sanctuaries. A wildlife sanctuary is a wildlife refuge, also called a wildlife sanctuary, is a naturally occurring sanctuary, such as an island, that provides protection for species from hunting, predation, or competition. It is protective area, that is a geographic territory within which wildlife is protected. Such wildlife regions are generally officially designated and designated territories. It is created by the government legislation, publicly or privately. कुछ एग्जाम्पल है, जैसे रूस में एक न्यूक्लियर एक्सीडेंट हुआ था. द चेरनोबिल न्यूक्लियर एक्सीडेंट साइट हैज एक्सीडेंटली बिकम ए वाइल्ड लाइफ रिफ्यूज बना दिया है उसको. India has 515 animal sanctuaries referred to as wildlife sanctuaries. Among these, the 41 tiger reserves are controlled, governed by the Project Tigers. तो करीब 500 से ज्यादा इंडिया में सेंचुरीज हैं, उनको यहाँ पे डॉट के रूप में दर्शाया गया है. इंडिया के हर क्षेत्र में है ये सेंचुरी, जैसे अभी मैंने आपको बताया बलिया में. This is Periyar Wildlife Sanctuary showing tiger. इसी तरह टाइगर रिजर्व है, जैसे पीलीभीत टाइगर रिजर्व, लखनऊ से आगे आप जाएंगे लखीमपुर खीरी के पास है, लखीमपुर खीरी. दुधवा नेशनल पार्क है उसके बगल में, और फिर आगे बढ़ेंगे तो कॉर्बेट नेशनल पार्क है. तो दुधवा तो और कॉर्बेट बेसिकली उत्तराखंड को ज्यादा कवर करते हैं. ये सारे टाइगर रिजर्व्स हैं. अब हम आते हैं एक्स सिटू कंजर्वेशन. तो सिंपल जैसे आपको समझ में आ गया कि इन सिटू कंजर्वेशन का मतलब है वहीं पे नेचुरल कंडीशन में ही जाके हम उन बायोडाइवर्सिटी को कंजर्व कर देंगे, जैसे प्लांट्स को, इन द फॉर्म ऑफ मेकिंग नेशनल पार्क, बायोस्फीयर रिजर्व, सेंचुरीज एंड अदर रिजर्व्स. अब बात आती है सेकंड पार्ट की, दैट इज एक्स सिटू कंजर्वेशन ऑफ प्लांट जेनेटिक रिसोर्सेज कैन बी अचीव्ड थ्रू डिफरेंट मेथड्स सच एज मेकिंग जीन सेंचुरीज, मतलब जेनेटिक मटेरियल को प्रिजर्व किया जाता है आइदर इन द फॉर्म ऑफ द सीड बैंक्स और फील्ड जीन बैंक्स. सेकेंड, इन विट्रो स्टोरेज मेथड, मतलब आर्टिफिशियल कंडीशंस में उस जर्म प्लाज्म को आइदर सीड के रूप में या डीएनए के रूप में प्रिजर्व किया जाता है. सेकेंड, पॉलिन बैंक्स, पॉलन ग्रेन्स को भी रखा जाता है. or DNA banks. Of gene sanctuaries. The genetic diversity is sometimes conserved under natural habitat. In other words, areas of great genetic diversity are protected from human interferences. Gene sanctuary is generally established in the center of the diversity or microcenter. India has set up its first gene sanctuary in the Garo Hill of Assam for wild relatives of the citrus plant. Now efforts are also being made to set up gene sanctuaries for banana, sugarcane, rice, and mango. There are some advantages of the gene sanctuaries. It protects the loss of genetic diversity caused by human intervention. It also allows natural selection and evolution to operate. Instead, there are few disadvantages. That is, entire variability of a crop species cannot be conserved. Its maintenance and establishment is a difficult task. मतलब costly affair है. Selection of superior germ plasm. Gene banks are also known as germ plasm bank. Gene bank refer to a place or organization where germ plasm can be conserved in living state, mainly in liquid nitrogen. Germ plasm are stored in the form of seeds, pollen grains, or in vitro cultures, or in the case of a field gene bank, as plant growing in the field. Gene banks are mainly of the two types, seed gene banks and field gene banks. First, seed gene banks. A place where germplasm is conserved in the form of seeds is called seed gene bank. Seeds are very convenient for storage because they occupy smaller space than whole plant. Seeds of all crops cannot be stored at low temperature in the seed banks. The germplasm of only orthodox species can be conserved in the seed banks. So in the seed bank, there are three types of conservation: short-term, medium-term, and long-term. Mainly depend upon the seed viability. Base collections are conserved for long-term, maybe 50 or more years at 18 to 20 degrees Celsius. So active collections are stored for short-term, 3 to 5 years at 5 to 10 degrees Celsius. Seed banks advantages. Large number of germplasm samples or entire variability can be conserved in a very small space. Second, in seed banks, handling of the germplasm is easy and cost effective. Third, germplasm is conserved under pathogen and insect-free environment. And about disadvantages, seeds of recalcitrant species cannot be stored in seed banks. Basically, जिनमें की viability बहुत कम होती है. Second, failure of power supply may lead to loss of seed viability and thereby loss of the germplasm in the form of the seeds. Third, it requires periodical evaluation of seed viability because after some time, multiplication is essential to get new or fresh seeds for storage. Second, field gene banks. Field gene banks or living collections are the main conservation strategy for long-lived perennial plants, recalcitrant species, and vegetative propagative species. Further, outbreeders require controlled pollination for regeneration from these seeds. These plants have an important educational value and can easily be assessed for research purposes. So field gene banks is one of the techniques or method in the conservation strategy for plant genetic conservation. It is an ex situ method where genetic material or genetic variation is maintained away from its original location, and samples of a species, subspecies, or even variety are transferred and conserved as living collections. Field gene banks is the most common method of conserving genetic resources with recalcitrant seeds and vegetatively propagated plants. There are few advantages as well as disadvantages. It provides opportunities for continuous evaluation for various economic characters. Second, it can be directly utilized in the breeding programs or plant breeding programs. There are few disadvantages. Field gene banks cannot cover the entire genetic diversity of a species. It can cover only few. That is a fraction of the full range of diversity of a species. The germplasm in field gene bank is exposed to pathogen and insects and sometimes is damaged by natural disasters such as bushfires, cyclones, and floods. So maintenance of germplasm in this field gene bank is costly affair than seed gene bank. Another method is in vitro storage method. The storage of germplasm in laboratory conditions or artificial conditions or in vitro conditions is especially suited for the long-term conservation of recalcitrant species or plant species and also vegetative propagative species. They can be stored at low temperatures under slow growth conditions or cryopreserved in liquid nitrogen at minus 196 degrees Celsius. Cryopreservation has so far been successful with only in relatively few plant species, but it is very promising development for long-term storage. The much limitation of the in vitro storage is the need for special equipment, techniques, and trained staff. However, more research is necessary to define the mechanisms of desiccation and chilling injury, and also to investigate methods for alleviating it. Pollen grain banks or pollen banks. Pollen preservation may useful for base collection of species that do not produce orthodox seeds, जिनमें regular seeds नहीं बनते, या percentage of viability is very low. It requires little space, but cytoplasmic genes would be lost. Like seeds, pollen can be divided into desiccation tolerant and intolerant pollens. However, information about the storage characteristics of pollen grains from wide species is fragmentary. That is existing mainly for some crop relatives only and for medicinal and forest species. DNA bank. The creation of a network for DNA bank is ex situ conservation method, and more precisely, germplasm collection can allow large quantities of genetic resources, mainly in the form of genes or DNA fragments, to be stored quickly and at low cost, and could act as an insurance policy against rapid loss of the world gene pool. It could be used in molecular phylogenetic and systematics of the extinct plant taxa, and genes can also be distributed via the polypeptide chain reaction using the primers, जो कि basically DNA primers कहलाते हैं, supplied by the users. So DNA samples have also been mainly used for bio prospecting and assessment of biodiversity studies. Its use in conservation is limited. As a whole, plant cannot be reconstituted from DNA, but the genetic material can be introduced to other genotypes for plant breeding and enhancement purposes. This example of botanical garden, जो कि situ का example है, conservation of endangered and threatened species. By making botanical garden, this is experimental garden in a controlled condition, such as glass house or greenhouses. We can maintain some important plants, especially by the use of tissue culture techniques. These are some of the references. So thank you very much for this lecture.